Hello students. So before we start discussing chapter 2, just a little piece of advice when listening to these recordings, and actually any other, by the way, would be to not listen to the whole hour of the lecture. Okay, that could get very boring. It's very easy to zoom out. So what you want to do is divide it into increments of maybe 10 or 15 minutes, take a break, and then come back and so on. So that's kind of what we're going to be doing during this chapter. So we'll talk about the chemical basis of life, which means we have to get into a little bit of chemistry here. So why is it that we're studying chemistry, although it's an anatomy and physiology course? Well, the thing is that a lot of the chemical reactions, or all of the chemical reactions really, that happen in the body involve the breakdown or the building of different chemical substances. Okay, and because they occur in the body, and we call that's a field of chemistry known as biochemistry, and it helps explain a lot of the physiological processes that happen in the body. And if there's an imbalance in these chemical reactions, we are going to call that a pathological condition or a disease. Okay, so simple things, or not simple, but normal things that happen in the body, like digestion, for instance, that breakdown of these bigger molecules into smaller molecules, that also involves biochemistry. The making of proteins, the building up of different proteins, that is biochemistry as well. So we really need to understand kind of like principles of chemistry, what organic molecules are versus inorganic, and so on. So first we'll talk about the structure of matter. Okay, matter is basically anything that takes up space and has a mass. That matter does not have to be a solid. Okay, it could be liquid or gas. And matter is made out of different elements. Elements are, if you remember, in high school chemistry class where we had this huge table of elements, okay, that had atomic weights and atomic numbers and so on. There were 98 of those elements that are found in nature. So a mix and match of these elements will give us different kinds of matter. The elements, we can break down elements into the smallest particle, and that is known as an atom. So the atom is the smallest particle of the element that has the property of that element. So I'll explain what that means. Meaning an H2, which is a hydrogen molecule, is the element of hydrogen, but we can break that down, that H2 molecule, into two hydrogen atoms, so H plus H. Now that hydrogen atom, the H, that is the smallest particle that has all the characteristics of hydrogen. If you break down that atom into the subatomic particles, like the nucleus and the protons, electrons, and so on, well, now these do not have any characteristics of hydrogen. So again, your atom is the smallest particle that has the characteristics of that element. Now, how much of elements do we need? There are lots of elements that our bodies need. We're not going to go into the details of how much of each, but we kind of categorize them into three categories. Some elements are needed in bulk amounts, okay, so kind of like Costco size and Sam's Club size, needed elements. We need those in large amounts, and those are carbon, oxygen, hydrogen, nitrogen, sulfur, and phosphorus. And you want to know these bulk elements. Trace elements, and the two that you want to be familiar with, are iron and iodine, and these are elements that we need but in smaller amounts. While ultra trace elements are required by the body but in very minute, very tiny amounts. Okay, for example, arsenic, we usually think of it kind of like a poison, which it is in higher amounts, but we do need these very minute amounts of it for certain chemical reactions. Now, as I mentioned, the atom is the simplest form of the element that keeps the characteristics of the element. If you break down the atom into its subatomic particles, we have lost those characteristics. So we'll take a look at this atom right here. This is an atom of lithium. And we can see here that it has a nucleus. The nucleus is made of these positively charged particles known as protons, and neutrally charged particles, or neutral particles, so they do not carry any charge. Those are known as neutrons. The surrounding the nucleus, we will find these negatively charged electrons that orbit the nucleus in these electron shells. These shells, the one that is closest to the nucleus is known as the least energy shell, followed by the one further away from the nucleus, and then we keep on going further and further away from the nucleus and so on. The one that is closest to the nucleus needs or can take up to a maximum of 2 electrons, while the following one can take up to a maximum of 8 electrons, and the third shell will be able to take up to a maximum of 8 electrons as well. So you kind of want to be familiar with those numbers, 2, 8, and 8. Now looking at the atomic structure, we could see here that the number of protons in this atom is equal to the number of electrons. A couple of things to keep in mind. First off, the only thing that is constant in atom would be the number of protons. So the number of protons in lithium is always going to be 3. Okay, that is known as the atomic number. While the number of electrons can be equal to the number of protons, as you see here, or they could be less or more. And that's why the atomic number, by definition, we say that the atomic number is the number of protons, but we do not by definition say that it's the number of electrons, because atoms have the ability to gain or lose electrons. Hence, the number of protons is always constant for an element, but the number of electrons can change. What about the number of neutrons? Number of neutrons can also change. So in this case, we do see that there are 4, but had there been 5 or 6 or 3, this would still be lithium because the number of protons are 3. So you kind of want to think of the number of protons as that social security number or the thumbprint of lithium. It will always be 3, no matter how many neutrons there are or electrons it has. As long as there are 3 protons in the nucleus, then that atom is a lithium atom. Okay, and that proton number is known as the atomic number. Here are, pretty much kind of what we talked about. We talked about the atom, the electron, proton, and neutron. So let's talk a little bit about an ion and a molecule. So an ion would be an atom that is either positively charged or negatively charged. And we'll talk about that a little bit more in the next couple of slides. So an ion is an atom that has an electrical charge. A molecule would be a chemical substance that is made of the union or the binding of 2 or more atoms together. Now moving on to the atomic number and atomic weight. The atomic number, as we mentioned, is the number of protons that are found in the nucleus. Okay, again, it is very unique to each atom. No matter what the number of electrons or neutrons are, you identify the atom by the number of protons. Again, that's the thumbprint or its social security number. Although the number of protons in a lot of times will be equal to the number of electrons, but by definition, we always count the number of protons as the atomic number because, again, the electrons can change. Atoms have the ability to lose or gain electrons, so the number of electrons is changeable, while the number of protons will always be constant. Now, the atomic weight or the atomic mass, that is really the weight of the nucleus. So if you go back to that picture, the atomic weight here you want to add up the protons plus the neutrons. So in this case, your atomic weight would be 7. For the atomic weight, we do not add the electrons. So the electrons really does not have a role or doesn't add anything to the weight of the atom. Now, what is an isotope? An isotope is an element that has the same atomic number, but has a different atomic weight. Meaning, so I'll give you an example. Oxygen, the atomic number of oxygen is 8. But we have different forms of isotopes of oxygen. So there's oxygen 16, 17, and 18. If the atomic number is 8, then where are these differences coming from? How is it that the atomic weight is different? That is due to the different numbers of neutrons. So the number of neutrons in here would be 8. The number of neutrons for the 17 would be 9, and the number of neutrons here would be 10. But because the number of protons in all 3 of these isotopes is 8, that makes all of these atoms an oxygen atom. Okay, so again, an isotope is the same atom, so they all have the same atomic number, but they differ in their atomic weights due to the difference in the number of neutrons. And some of them are radioactive, so we can use that radioactivity to detect, diagnose, and sometimes even treat different conditions. So we'll take a look at an example right here, and this is the the use of the radioactive isotope iodine-131. The thyroid tissue, or the thyroid gland, is an endocrine gland that releases a hormone, or hormones. It's found in the front of the neck, and it is the only tissue in the body that uses iodine. So we can give patients radioactive iodine that will be picked up by thyroid tissue, and then scan the body and see if there's any activity, any radioactive activity. That means that there is thyroid tissue in that area. So we'll see here, this is what a normal thyroid gland looks like. While in this patient, you can see here that there's a lot of uptake in the amount of iodine, and that means this patient has Graves' disease, which is a form of hyperthyroidism. So their thyroid glands are hyperactive, and we were able to detect that because of the increase in the amount of uptake of the radioactive iodine. In this image here, we could see that there's uptake of radioactive iodine by the bones, right here in the femur, for example, in the spine, in the pelvic bone, and so on. So this indicates that this patient has thyroid cancer that has metastasized to the bone. So remember again, thyroid tissue is the only tissue that can actually utilize iodine. So since now we can see that that iodine has been taken up by bone tissue, that means that there is metastatic thyroid tissue found in those areas. We can use to detect metastasis, and we can also use it as a follow-up. Let's say this patient takes chemotherapy, radiotherapy, or whatever kind of therapies they'll go through. We could reassess, say, maybe a month or two later and see if these lesions are growing or are they responding to treatment. So we can use these radioactive isotopes to either diagnose or evaluate the efficiency of treatment. Another thing we use is known as PET imaging, or positron emission tomography. And these are very unique kinds of isotopes because they emit something known as a positron. A positron is an electron that is positively charged. So remember, electrons are negatively charged. But in these cases, this radioactive isotope is emitting a positively charged electron, hence the name positron. And we usually use these to detect different, blood flow, especially in the brain, and to kind of better understand brain physiology. Now we'll talk about molecules and molecular formulas. So a molecule, as we mentioned before, is the combination of two or more atoms together. Okay, if you combine two or more atoms chemically, we have now made a molecule. If those atoms are the same, we are going to call that an element. If the atoms are different, we'll call that a compound. So a molecule is kind of an umbrella that encompasses both elements and compounds. So I'll come back to this in a minute, but I want to show you the difference between an element and a compound. So right here we have two hydrogen atoms combined together. That is a hydrogen molecule for sure, and an element because both hydrogen atoms are the same. Same thing with oxygen right here. But if you look at the water molecule, it is made out of oxygen and two hydrogen atoms. So now that we have two different kinds of atoms, this makes it a compound. Again, remember that all of these are molecules because we have two or more atoms combined. And underneath that umbrella of molecule, we have elements if the atoms are the same, or compounds if the atoms are different. So what about these molecular formulas that we sometimes read? Well, it just gives us an idea about what the atoms are in each molecule and how many of each. So we'll use this hydrogen molecule H2, which means it's made out of 2 hydrogen atoms. The molecule of glucose, C6H12O6, which means it's made out of 6 atoms of carbon, 12 atoms of hydrogen, and 6 atoms of oxygen. And then the molecule of water, which is the infamous H2O. So which one out of these is an element? If you answered hydrogen, then you are correct, while the other two, glucose and water, are compounds. Now, which one of these are molecules? They are all molecules. So again, molecule encompasses both elements and compounds. Okay, so we kind of already went over this, the element versus compound. Now this would be a very good time to stretch, take a break, jumping jacks, whatever it takes in order to be able to focus on what we're going to be talking about in a bit, and I'll take a break as well. Okay, so welcome back, and next we'll talk about bonding of atoms. So now we know what atoms are, and we know that they have to combine together to make different molecules. Now, how do atoms bond? Most of the bonding happens because of the action of electrons. And we already kind of touched bases on the shells and the orbits where electrons live. But since it's up here, we'll talk about them again. So the electrons go in these orbits, and these orbits are known as electron shells or energy shells. The very first shell, which is the lowest energy shell, can hold up to 2 electrons. The second shell can hold up to 8, while the third shell can also hold up to 8. As you get further away from the nucleus, these orbits are obviously bigger. They can hold more electrons. Some can even hold up to 18 electrons, and those are usually in the bigger atoms. Keep in mind that in order for an atom to be stable, you want the shell to be saturated with electrons. So what on earth does that mean? We'll kind of discuss that using the sodium atom as an example. Okay, so you'll see the sodium atom with 2 electrons on the first shell, 8 electrons on the second shell, but the third shell has 1 electron, meaning that this is an unstable atom. In order to become stable, you either want to add 7 electrons to that outermost shell or remove that 1 electron so that the second shell will be saturated with all 8 electrons. And that's really what the sodium atom does. It gets rid of that extra electron, and because it lost an electron, sodium becomes a positively charged ion. We call a positively charged ion, we call that a cation. And in order for me to remember positive versus negative, cats kind of give you that positive vibe, so cations are your positively charged ion. And again, it is formed when an atom loses an electron. Now, if you compare that to this chlorine atom, you will see that the outermost shell has 7 electrons. So again, it is an unstable atom. In order to stabilize, it needs to fill out that shell to the max of 8. So it will take that extra electron, and now this outer shell would have 8 electrons, and this now chloride ion becomes stable. Because it gained an electron, which is negatively charged, a chloride ion is negatively charged as well, and we call that an anion. Okay, so In short, ions are atoms that are either positively charged, we call those cations, or negatively charged, and we call those anions. And it happens due to either the loss or the gain of an electron. Now, the first kind of bonding is known as an ionic bond, where this positively charged ion will attract the negatively charged chloride. Okay, so remember rule of physics that positive and negatives attract. So that is the force that causes that ionic bonding. Okay, so again, the anionic bonding is the bonding between a cation and an anion due to the electrical charges. It is a very strong bond, and in this case we're talking about the bond that forms sodium chloride, which is salt, table salt. Okay, now next to the second kind of bonding that we'll talk about are covalent bonds. Covalent bonds are also due to electrons, but in this case, there is no loss or gain of electrons. Rather, there is sharing of electrons. So you kind of want to think of covalent kind of like co-workers, colleagues that are working together, and they share the electrons with each other. These are one of the strongest chemical bonds that are found that we're going to be talking about. So you can see here a hydrogen atom and another hydrogen atom. Each one of these has a little single electron in the shell. And in order to stabilize, it would need two. So it would need to gain an electron. So with this one, so what they do is they share their electrons. So now you can see here that they are sharing their electrons together, and they are sharing it equally. Okay, so now, and this sharing of electrons is known as covalent bonds. Here you can see again, the sharing of electrons between these hydrogen atoms. Same here with these two hydrogen atoms. Here in these two oxygen atoms are also sharing, but you'll see here that they are sharing two from each, so a total of 4 electrons, making this oxygen molecule. If you look at this water molecule, you'll see that there is a sharing of electrons between the oxygen and hydrogen. But because oxygen is bigger, much stronger than these two hydrogen atoms, there is unequal sharing of electrons. So the oxygen, being the bigger of these atoms, will pull those shared electrons towards itself, making the oxygen end a little bit negative, while the hydrogen ends would be a little bit positive. And this is known as polar covalent bonds. So polar means having a positive pole and a negative pole, kind of like a battery. And that happens in these polar covalent bonds. So what we see here is these two atoms have the same strength, they're the same size, so they share the electrons equally. This is an example of a non-polar covalent bond. We do not have a positive and negative end. But in the case of a water molecule, the oxygen is bigger and stronger and pulls the electrons towards itself further away from the hydrogen. So they are still sharing the electrons, but the sharing is unequal. So you'd have a negative end towards the oxygen and positive ends towards the hydrogens. And again, we call that a polar covalent bond. So what are these? And I'll come back to this in a minute. I just want to, finish this point about these polar molecules. So a polar molecule would be a molecule that has a slightly negative end and a slightly positive end. And we're taking a look again at this Mickey Mouse looking water molecule. And again, it's due to the unequal sharing of the electrons in these covalent bonds. These polar molecules are extremely important because they can lead to what are known as hydrogen bonds between water molecules. And I'll show you that image right here. And again, I'll go definitely back to that one slide, that one of the weakest kind of bonds that we have in the body are known as hydrogen bonds, where the negative end of oxygen is attracted to the positive end of hydrogen. So although the water molecule as a whole is neutral, but again it has a negative end and a slightly positive end. So again, negative and positives attract, leading to these very weak hydrogen bonds. And you see the weakness indicated by these dotted lines. These hydrogen bonds are extremely important when we make proteins, and it's also important in the structure of nucleic acid, and we'll talk about that towards the end of this chapter. So I want to go back to the structural formulas and kind of comparing that dotted line that we've seen in the other slide to these solid lines. So that one solid line represents a strong covalent bond. And because it's only one line, that means that only one electron, there's only one bond. There is sharing of one electron between these two atoms. If we look at this oxygen right here, again, there are solid lines, so these are strong covalent bonds. There are two of them. That means they are sharing 2 electrons each, so there are 2 bonds between the oxygen atoms. And you can kind of go through these examples on your own. So again, you want to understand the difference between these solid covalent bonds and these interrupted hydrogen bonds. Okay, so we already talked about these. Now a little bit about chemical reactions. So how do these different chemicals react together? And a chemical reaction would be basically breaking down bonds and making new ones. And in any chemical reaction, we have something that starts the reaction. We call that the reactant or the substrate. In this example, it would be sodium chloride. And here we are breaking down that bond in order to make your end products. And the end product here would be a positive sodium ion and a negative chloride ion. This breaking down of a bigger molecule into smaller components is known as a decomposition reaction. Okay, and this can occur in water. So when we dissolve salt in water, we are breaking down the sodium chloride molecule into sodium and chloride ions. And because we have positive and negative charges, that water can now conduct electricity, and hence the name electrolytes. That's where the name comes from. So that was an example of a decomposition reaction, okay, where we broke down the sodium chloride into the components of sodium and chloride. A and B are just like symbols, like, you know, if you remember in algebra, x plus y equals whatever. So these are just symbols that we're using. They're not actual real chemical compounds. So that's one example of reaction. Another example of reaction would be synthesis reactions. So we are building up a bigger molecule. We take the smaller components or the building blocks and we build up a more complex chemical structure. Most reactions in the body are reversible. Okay, so I'll go back to the sodium chloride example. So we could break down sodium chloride into sodium and chloride ions, or it could be reversed back into a sodium chloride molecule. And that is known as a reversible reaction, okay, where the products can change back to the reactants. So we could either synthesize or decompose. And again, most of the reactions in the body are reversible. An exchange reaction is where we break chemical bonds and we make new ones. You want to think of exchange reaction kind of like you have two couples that are dancing, and then they exchange their dance partner. That's exactly what happens in an exchange reaction, where you have this bigger molecule of AB and another molecule of CD. We are going to break the bond between A and B and break this bond between C and D and then exchange. So we'll make new bonds between A and D and B and C. Okay, so again, you are breaking chemical bonds in order to make new ones. So what is the difference between acids, bases, and salts? We kind of already talked about electrolytes, and that would be anything that can dissolve in water and end up making a positive and negative ion, or making positive and negatively charged ions. Okay, so what is an acid? An acid by definition is an electrolyte, so it has to be something that can dissolve in water, and it will dissociate into a positive and negatively charged ions. One of the positively charged ions has to be hydrogen. Okay, so an acid is an electrolyte that dissolves to release hydrogen ions in water. The more hydrogen ions released, the stronger that acid is. While a base is something that is going to also release ions, one of those ions can react with hydrogen to make water. So when I was in college, basically bases were, by definition, a substance that can release a hydroxyl group. So this OH group is known as a hydroxyl group. That has changed over the years, and they changed the definition to where a base is a substance that releases an ion that combines with hydrogen to make water. A salt is an electrolyte that is formed by the reaction between both of these, between an acid and a base. So if you bring in hydrochloric acid with sodium hydroxide, and by an exchange reaction where we break the bond between the hydrogen and the chloride, we break the bond here between the sodium and the hydroxyl group, we end up making that salt of sodium chloride and removing a water molecule. So that's how a salt is made, by a reaction between an acid and a base. Now we do have different acids in the body. For example, gastric juice has hydrochloric acid, which is extremely acidic. We need to make sure that the amount of acid in our body remains within normal range. And we measure that through a scale known as the pH scale. The pH scale is kind of like a complicated algorithm, but it indicates the concentration of hydrogen ions in a solution. The scale ranges from 0 to 14, where 0 is the most acidic and 14 is the most basic or alkalinic. Right in the middle is neutral, and that would be distilled water. Okay, anything between 7 and 0, or less than 7, all the way down to 0, is acidic. Anything between 7 and 14 will become alkalinic. So if you take a look at these, you don't have to know these numbers or anything. It's just kind of interesting to look at. For example, apple juice has a pH of 3, while tomato juice has a pH of 4.2. So they're both very acidic solutions. Gastric juice, on the other hand, has a pH of 2 because of the hydrochloric acid. You will see, though, that the human blood has a pH of 7.4. There's a range, really, 7.35 to 7.45. And so how is it that although our bodies make that very acidic hydrochloric acid, how is it that human blood remains at, within that range of 7.4, which is actually on the alkalinic side? And we will talk a lot more about this when we get into the respiratory system and the renal system. So don't worry about it for now. So again, that normal pH range, though, is something that you want to know right now, and that is, again, that those numbers range from 7.35 to 7.45. Keeping, though, the pH within normal range is known as homeostasis. Okay, so we have like these different buffers and chemicals that, again, we'll talk in more detail about when we get to the respiratory system and the renal system. But just know for now that through homeostasis, we have different chemicals known as buffers that resist any change or drastic change to the pH. But do we really always remain within that normal value? Unfortunately not, because just like anything else in the body, sometimes we succeed in keeping it within normal, sometimes we don't, and we end up going through either acidosis or alkalosis. So acidosis would be if the pH ranges between 7.3 to 7.0. Anything which is, remember, that was your neutral. Anything below that is really kind of incompatible with life, so you want to keep that range. We're already sick through this acidotic range where the person is disoriented, they're fatigued, they feel tired. And we can see that in patients, for example, with diabetic ketoacidosis, where if there's not enough insulin in the body and they're unable to utilize glucose, that glucose is going to change into ketone bodies, which are basically acids, and that can lead to a condition known as diabetic ketoacidosis. Lung diseases is another way of developing acidosis because due to the inability to get rid of carbon dioxide. So know that carbon dioxide in the body can combine with water to make an acid known as carbonic acid, or H2CO3. Please keep that somewhere within your head, because it's going to haunt us again when we talk about blood, when we talk about the respiratory system, and maybe even the renal system. Okay, so keep this important piece of info somewhere, that carbon dioxide in our bloodstream dissolves or combines with water and they make carbonic acid, which means that if we accumulate carbon dioxide in our bodies, we are ending up making a lot of carbonic acid, and that's why we end up going through acidosis. So if a person, for instance, has severe pneumonia and they're unable to get rid of all of that carbon dioxide, there will be a buildup of carbonic acid leading to acidosis. Okay, now, so moving on to alkalosis. Okay, alkalosis is a pH of the blood between 7.5 to 7.8. In this case, patients feel dizzy, they feel agitated, anxious, and this could happen anything that can lead to, for example, hyperventilation. So having maybe a panic attack or anxiety, that being in high altitudes. Okay, so that hyperventilation, remember again, you're washing away a lot of that carbon dioxide, which basically means you're getting rid of a lot of carbonic acid, which means you're leaving your body with not enough acid. So that it, relatively speaking, it becomes an you go through alkalosis. If there is severe vomiting of all of that gastric juice, okay, so you've lost, again, a lot of acid, your body goes through alkalosis. Sometimes it's induced. So patients might have, severe heartburn or acid reflux, and they self-medicate with a lot of antacids. Most of these antacids are alkaline, and they are kind of self-inducing, introducing all of that alkaline or all of that base into their body, so they induce that state of alkalosis. Again, our body has some homeostatic mechanisms to work on this and try to correct it. If the different chemicals are called buffers, and again, we'll go through these in more details later on. So now again is a very good time to go stretch, de-stress, and come back. Okay, welcome back, folks. Now we will talk, now that we've learned about atoms, different chemical reactions, we want to talk about the chemical constituents of cells. So we're talking about organic versus inorganic molecules. And we kind of all, hear the term all the time. But what are organic molecules? An organic molecule are molecules that have lots of carbon and lots of hydrogen. Okay, so if I ask you, CO2, carbon dioxide, is that an organic molecule? It is not, okay? It has to contain a lot of carbon and a lot of hydrogen in order to become organic. So you will see that carbon dioxide here is an inorganic molecule. And some of these organic molecules, some of them can dissolve in water, like carbohydrates. Okay, so sugar, glucose, for example, you can definitely dissolve that in water. Others, on the other hand, like lipids or fats, can dissolve in organic liquids, which would be basically dissolving in other oils. Okay, so we'll find that some organic molecules, some can dissolve in water, others can dissolve in oils. The water-soluble organic ones are not going to release ions, and they are not electrolytes. So if you dissolve sugar in water, you are not making positively and negatively charged ions. And we are not going to call sugar an electrolyte. Okay, so compare that to the sodium chloride that we just talked about in the last couple of slides. Now, the organic molecules that we are going to be discussing are carbohydrates, proteins, lipids, and nucleic acids. But before we do so, we're going to talk about the inorganic molecules, and these generally do not have carbon or hydrogen. And those can dissolve in water. They are going to form ions and are considered electrolytes. Examples of inorganic molecules are water, oxygen, carbon dioxide, and inorganic salts, for example, sodium chloride, potassium chloride, magnesium sulfate. All of these are considered inorganic salts. Okay, so just a quick question here is, so we talked about carbon dioxide being an inorganic molecule. It still has carbon, okay, but it has a few, just one atom of carbon, and so hence we'll consider it inorganic as opposed to organic. And when we look at the structure of the organic molecules, you'll see that it has a tremendous amount of carbon and hydrogen. So we'll talk a little bit more in detail about these inorganic substances. First off, water. And we are 2/3 by weight water. Okay, which is great. So whenever you go on that scale and you take a look at that number, know that 2/3 of that number is actually water. And water is extremely important because this is where all of the chemical reactions will occur. We need water we'll find out that plasma is about 90% water. And we need that water so that things can circulate throughout the body. Things like oxygen, carbon dioxide, glucose, hormones, enzymes, all of these won't be able to circulate throughout our bloodstream if there was no water there. And it is also important in the regulation of body temperature. Okay. Oxygen is necessary for survival, and oxygen is used by organelles, for example, the mitochondria, and in order to release energy. So when we burn glucose, for example, some of the steps are going to need oxygen in order to make bigger amounts of ATP, or the molecule of energy. Carbon dioxide is actually considered a waste product, and due to the metabolic reactions that occur in the body, one of the waste products that we make is carbon dioxide, and we have to exhale that. We have to breathe it out in order to get rid of it. Remember that carbon dioxide will combine with water to make carbonic acid. Okay, so that could lead to acidosis, and we have to get rid of that waste product. Now, inorganic salts, different kinds of them, sodium, chloride, potassium, calcium, all of these different ions. And we will talk about the use of these ions and get into more detail. So, for example, we will find that calcium is extremely important in muscle contraction. Sodium, chloride, and potassium, for instance, are extremely important when it comes to the conduction of electricity on nerves. Potassium is also, and calcium are also, both of these are really important when it comes to blood clotting. So all of these inorganic salts have there's distinct roles that we'll talk about as we get to each individual system. Now for the organic substances, and the first one we'll talk about are carbs. That is the main source of cellular energy. Okay, it is really important because it supplies the material to build cell structures. It supplies the energy to do those, and carbs are water soluble. They contain carbon, hydrogen, and oxygen. The ratio between hydrogen and oxygen is very close to 2:1. So we'll see in this glucose molecule, C6H12O6, the ratio between hydrogen and oxygen is actually exactly 2:1. And you want to keep that in your mind, because when we compare it to lipids, lipids also have carbon, hydrogen, and oxygen, but the ratio between hydrogen and oxygen is not going to be or even close to 2:1. Okay, so if you see a structure of formula and you're not sure if it's a carb or a lipid, you could look at the ratio between hydrogen and oxygen. If it's equal to or very close to 2 to 1, that would be a carb. If it's not, then it's a lipid. So how do we classify these carbs? Well, we classify them, I'm actually going to go backwards. We classify them according to these bigger complex carbohydrates. Those are known as polysaccharides. So saccharide, the word saccharide means sugar. Poly means a lot. So we have these several building blocks of sugar to make these complex carbohydrates. For example, starch, glycogen, and cellulose. Glycogen is how the body stores glucose. We store glycogen in the liver and in our skeletal muscles. If you're unfamiliar with skeletal muscles, that's fine. But in short, these are the muscles that attach to the skeleton and help us move. Hence the name skeletal muscle. And again, we store glycogen in the muscle and in those muscles and in our liver. And we are going to use that stored glycogen when we're in between meals in order to utilize that for energy. Okay, as we eat starch, okay, we have to break it down. Our intestinal cells are unable to absorb that huge complex molecule of starch. The body, the digestive system, has to break it down into smaller molecules. So the first step would be to break it down into these disaccharides. So di is 2, saccharide is sugar. So these sugars, these sucrose and lactose, only have 2 sugar units attached to each other. And one of the last steps, or the last step of digestion of carbs would be the breakdown of these disaccharides in the cells of the intestine, of the small intestine, using an enzyme. For example, sucrose will break down by an enzyme known as sucrase, while lactose, we will break that down with using an enzyme known as lactase. Some individuals do not have the lactase enzyme, so they are not going to be able to break down that lactose, which comes from milk products, into its monosaccharides or building blocks, which are galactose and glucose. So what happens is that there will be an accumulation of that lactose inside of the small intestine. It will remain there, and being a sugar, it will ferment over time, and it will lead to the release of a lot of gas, leading to distension, cramps, diarrhea, and so on. And the treatment would be the intake, or to decrease the intake, rather, of lactose, so decrease the intake of milk products. And sometimes in more severe cases, patients would have to take a lactase tablet in order to help digestion of any lactose. The sugars that our intestines are able to absorb would be these monosaccharides. So these are made out of just one single sugar, and those are your glucose and fructose. Okay, those are just examples. There might be more. So here you can see what they look like. So this is your polysaccharide that, again, we might eat, but we're unable to digest. We're unable to absorb. So we'd have to break it down and digest it into this disaccharide. And then the last stage of digestion would be to break this disaccharide into monosaccharides. And that happens in the intestinal cells of the small intestine. And these monosaccharides is now what we can actually absorb. The second organic substance to discuss would be lipids. Now, lipids or fats are insoluble in water, but they are soluble in organic solvents, which are basically other lipids or other oils. That includes triglycerides, phospholipids, and steroids. So we'll talk about all 3 of them, and we will see that when we talk in chapter 3, we'll find that all 3 of these are extremely important components of the cell membrane. And triglyceride is just a fancier term for fat cells. Okay, fat cells, and that's what we'll talk about first, or triglycerides. I just want to show you what they look like first. They are actually a molecule of glycerol, so that glycerol portion, with 3 fatty acid chains attached to it. Hence the name triglyceride. So 3 fatty acid chains attached to that glycerol molecule. And it can be used for cellular energy, and it actually stores more energy than carbs do. The only thing is that, in between meals, so let's say somebody's going maybe on a diet, okay, and let's do these intermittent fasting diets, okay? So the first few hours after a meal, after the last meal, the patient or the person is going to start tapping into their storage of glycogen first. So they'll use their glycogen storage in the liver, skeletal muscles. When those are depleted, the next step would be to tap into the triglyceride or the fat cells, okay, in order to burn that for energy. Triglycerides, as we mentioned before, has carbon, hydrogen, and oxygen. But again, that ratio is not 2-to-1. The amount of oxygen is a lot less. And we already mentioned that it's made out of a molecule of glycerol and those 3 fatty acid chains. So looking at the fatty acids, we have 2 different kinds. They're saturated or unsaturated. A saturated fatty acid, like this one up here, you will see that all of these carbon atoms are saturated with hydrogen, and each carbon atom is attached to the next one or its neighbor with just one covalent bond. Okay, these are usually at room temperature. These will solidify, and these are of animal origin. While the unsaturated fatty acids, you'll see here that not all carbons are saturated with hydrogen. Rather, some of the carbon atoms have to have more than one covalent bond together. These are of plant origin, and these are liquid at room temperature. Now, which one do you think is the healthier option? Well, the healthier option are definitely these unsaturated fatty acids, so the plant origin ones, so like olive oil, corn oil, and so on. Now for lipids. Okay, sorry about that. I had to put the charger on. It'll die, and I can't have that happening after all that work. Okay, next thing on the list are phospholipids. So, going back to the composition or what a triglyceride molecule looks like, remember we said it has a glycerol portion and 3 fatty acid chains. Well, if you compare that to a phospholipid, what happens in order to make a phospholipid is that we are going to add a phosphate group to one of the fatty acid chains. Okay, so we'll add that phosphate group, the PO3, okay, and keep the other 2 fatty acid chains as they are, and hence the name phospholipid, because we have that lipid and we added the phosphate portion. Now, a triglyceride is totally insoluble in water, okay? It is hydrophobic, it's afraid of water, while phospholipid is a little bit different. So because we've added that phosphate portion, that will give us this water-soluble head, or the hydrophilic head, while we still have these 2 fatty acid chains that will give us these 2 tails of hydrophobic tails, or water-insoluble tails. When we get to chapter 3, you will learn that a cell membrane is made out of 2 layers of phospholipids, and it is the structure of having that hydrophilic head and 2 hydrophobic tails that is extremely important in regulating what can get in and out of the cell. Now going on to steroids, which is another kind of lipid. Steroids are derived out of these 4 rings of carbon. All 4 rings are connected together, and at each angle you'll find a carbon ion, or carbon atom, I'm sorry. You can see them all here with all of the hydrogen attached to it. Do not really worry about the composition of cholesterol, okay, but know that cholesterol is a steroid, and it is the building what we use in order to build a lot of hormones, so we have what are known as steroid hormones. For example, the sex hormones like estrogen, progesterone, and testosterone, those are cholesterol based. We also have hormones coming out of the adrenal cortex like cortisol, for example. These are also steroid hormones, and they're also derived out of cholesterol. So we kind of think of cholesterol as, that bad thing, but which it is in higher amounts, but we still need some cholesterol in order to build these steroid-based hormones. And we'll also find out that it is a component of the cell membrane. It is a little bit, quote unquote, harder than phospholipids, so it gives it a little bit of the cell membrane a little bit of a structure. Now for proteins as our third organic substance, so we discussed carbs and lipids, now we'll discuss proteins. Proteins are really, the instructions to make proteins are found in our DNA, in the genes. Okay, and we pretty much make proteins for everything. So our muscles are made out of proteins. Hormones, we have lots of protein hormones, antibodies, enzymes. All of these are examples of proteins that our body makes. We also have what are known as plasma proteins. These are proteins that are made by the liver, and they circulate in the plasma in our blood. They are responsible, we call them colloids, and they're responsible for a kind of pressure known as the colloidal pressure. And again, keep that somewhere written down, and we were going to need this when we talk about urine formation by the kidney. Okay, the colloidal pressure of proteins is an important fact, kind of helps us in doing that. Proteins are made out of building blocks known as amino acids, and you can see here that there is one amino acid right here, and this is the second amino acid, and then there's this red bond in between, and that bond is known as a peptide bond, which is a bond kind of unique to the building of proteins. Now proteins, as I said, we have the instructions in our DNA. Okay, and the proteins, some proteins are very kind of complicated in their structure. Okay, so there are different levels of making proteins. There is a primary level, which is, all proteins have to have this primary level at least, where it detects the sequence of amino acids. So how do these amino acids get in order? What is the exact order of these amino acids? And that is what is found within the DNA. Okay, so the primary structure of protein would be basically making this bead of amino acids and letting us know the exact sequence of those amino acids. And again, remember that they attach to each other through what are known as peptide bonds. The secondary structure, okay, we know that some of these proteins are really long chains, and you don't want these chains circulating within the bloodstream. You kind of want to pack them a little bit. So we can move on to a secondary level of protein structure. So after making that primary sequence, we can either fold them like a fan. Okay, that would be known as a beta pleated sheet, or we can twist them, and that is known as an alpha helix. That twisting or folding are made by hydrogen bonds found between non-adjacent amino acids. So let's explain this kind of like in English. So the primary structure would be peptide bonds between the adjacent amino acids, so the ones that are neighboring to each other, while the folding part, whether pleated or twisting, happens because of hydrogen bonds happening, say, between these two amino acids. So they are not adjacent to each other, they're not neighbors. Okay, so these are the adjacent two. These two are not adjacent, and hydrogen bonding will occur between them, leading to either the folding or the twisting. The tertiary structure, we are going to pack them into a 3D fold of a protein. Most proteins really end at this tertiary structure. There are some very globular, very complicated proteins, bigger molecules like hemoglobin, for example, where we are going to go to the fourth level or quaternary structure, where we'll take two or more of these and attach them together. So you see here we have 1, 2, 3, 4 of these combined together to make this huge molecule. Now for the last of the organic substances, which are nucleic acids, and nucleic acids are either your DNA or RNA. DNA, that carries the genetic code and it's found within the nucleus, while RNA helps in protein synthesis. So we do have some RNA in the nucleus and some RNA in the cytoplasm out of the nucleus. As we said, already mentioned, that the DNA that can encode or has the instructions, has the coding for the amino acid sequence of the proteins. The building block of nucleic acid are known as nucleotides, and here is kind of a depiction of what a nucleotide would look like. There is a sugar, a phosphate group, and an organic base. So the sugar in DNA is called deoxyribose, hence the name DNA or deoxyribonucleic acid, while the sugar in RNA is known as ribose sugar, and that's where the name ribonucleic acid comes from. The phosphate groups are the same, while the organic bases are different. So in DNA, the organic bases can be 1 out of 4, and I'll just go to the list so you can see them in writing. They can be cytosine and guanine, or adenine and thymine. And I kind of paired them this way because they fit together like a puzzle. So cytosine always has to attach or bond with guanine, and adenine always bonds with thymine. And we'll go back to that image and kind of understand what that means in a minute. While RNA, on the other hand, can have 1 out of 4 bases as well, so cytosine and guanine and adenine. So these 3 are shared between DNA and RNA, but RNA does not have thymine. It rather has uracil. So if you find a structure that has uracil in it, it has to be RNA, while a structure that has thymine in it would have to be DNA. So we'll go back here to to look at the differences between RNA versus DNA. If you look at RNA, it is made out of a single chain of nucleotides. The single chain has, there's one nucleotide right there, the sugar, ribose sugar, phosphate group, and a base that can again be either cytosine, guanine, adenine, or uracil. And then there is a bond between the sugar and the phosphate group, and that would give you your single strand of RNA. While in the DNA, it is a double strand. So there's one strand on this side, and there's your second strand. And they join together through these bases. And this is where the cytosine and guanine, or adenine and thymine, come together. So again, cytosine and guanine, those are going to hold hands, while adenine and thymine will connect. Now hopefully you'll be able to understand by this point what these dotted lines mean. So what kind of bond do we have between these bases? These bonds are the weak hydrogen bonds, and that is why if a person gets a very high temperature, these hydrogen bonds can easily break. Now notice this is your DNA, okay? So if we break the DNA, we break the genetic code, that is basically killing those cells. Okay, so I believe this, and it does concludes chapter 2. Please let me know if you have any questions.